do we start with genetically modified foods? I mean, we've already sort of started talking about it, but
yeah. Let's start with the basic concept that humans have been genetically modifying their food for 12,000 years.
Yes, and this is something that we forget. There was the whole idea of grafting plants together to combine their qualities. I have a tree in my front yard, Richard.
Yeah?
It's a cherry tree, but it's two trees. It's one with pink flowers on top of one with white flowers. And
nice.
I never noticed it until a couple years in. I'm like, hey, there's two trees in this tree. And it's very simply, one was spliced onto the other one and they just fused together.
Well, and in fact, virtually every fruit tree you've ever seen was a graft.
Yeah.
Because the genetics of, say, apples are so unstable that if you take the seeds from a red delicious apple and plant them, you will not get red delicious apple trees. Right? They're not genetically stable enough. The tree you grow will probably be small apples that are bitter. And so, in fact, every red delicious, the reason it has that name, comes from the same tree.
Right.
The normal practice in fruit trees is that you cut away small sapling branches and you graft them onto other trees.
So we have literally made fruits and vegetables in the image that we wanted them to be.
Right. And I just sent you a link, and I'll include this in the show notes. It is a collection of pictures of what fruits and vegetables look like in their original form. So it's just this idea of the wild watermelon was almost all the white part with a little bit of seed. The red part in watermelon, that's actually placenta.
And you can tell because they show a painting by Giovanni Stanchi, or Stanchi, Stanchi, from between 1645 and 1672 of a watermelon. And it looks nothing like a watermelon. It's all pith.
We think of today.
Yeah, it's all pith.
And same with the bananas. Normal, original bananas had huge seeds on them. The modern banana, which is dying out because of a blight,
right,
has so few seeds that almost a third of its crop go into trying to find enough seeds to keep planting it.
Yeah.
Because we've bred them to the point where they basically don't make seeds anymore. And it goes on from here. You know, carrots were originally white. The beta carotene that we now name carrots for, we evolved that trait in. Well, one of my favorite sets of vegetables to look at are the Brassica oryxaris, right?
And what is that?
That's the wild cabbage. So we see evidence that humans were selectively breeding that plant on the coast of the Baltic when they were still hunter-gatherers.
Wow.
Right? They would make camps along the Baltic Sea in the summertime, and they would collapse them in the wintertime when they left, but they were picking certain plants and keeping them in the same location. So they encouraged the large-headed cabbage to first come along. We've got evidence that it's, in the BC, but that same plant is responsible for cabbage, collard greens, kale, Brussels sprouts, broccoli, cauliflower, broccolini. It's all the same plant.
Right.
We've just been selectively breeding it for thousands of years.
Humans like coleslaw, apparently.
Yeah. Pliny the Elder wrote about the merits of cauliflower.
We wouldn't have a show on food without talking about Pliny the Elder. He comes up all the time in conversation. It's really true. I mean, the more you start looking around on the internet, the more he comes up. He's like central to this story.
It's very funny.
Yeah.
Have you ever heard of a plant? It's a Chinese broccoli called gailan.
No.
Very popular here, but it's the same plant. And since the brassica is from Western Europe, it's sort of evidence that the Silk Road was a Silk Road way before it was called the Silk Road.
Okay.
That some version of broccoli made it all the way east and became a popular food.
Well, cabbage is a wonderful food for trade because it transports so well.
Absolutely. And that turns out to be a very big issue.
Yeah.
So selective breeding's gone on a long time, and nobody is afraid of selective breeding, although maybe they should be. We've never done genetic engineering of chickens in production.
Yeah.
Okay. And just with selective breeding, what we've done to chickens is kind of horrifying.
And we talked about this before, didn't we?
I don't think we've done it on the show, but it's certainly, I gathered this data a while ago because I was trying to get a comparison of what's happened to chicken. I mean, basically, industrial farming now makes two kinds of chicken. The food chicken, right? They call the fryer, and the egg-laying chicken.
Yeah.
Now, by selective breeding alone, We've gotten egg layers to the point where they used to put out about 30 to 50 eggs a year 100 years ago. They now put out 300 eggs a year.
Wow.
And by the way, when you're breeding egg-laying chickens, you only want the females, right?
Right.
Don't talk to me about what happens to male chicks when they're from the egg-laying species. It's as horrifying as you can think of.
Yeah.
And on the food side of chickens, in 1957, a newborn chick weighed 34 grams, like an ounce and a half. And in 60 days, it was up to about 2 pounds, although it really took another 60 days to be fully grown. By 1978, in 60 days, it was 4 pounds. So they doubled the weight of the chicken in the same amount of time. By 2005, we got it up to almost 8 pounds. We doubled it again. And mostly in the form of breast meat, to the point where about 25% of the birds, when they're ready for harvest, can't even walk.
And is this done by somebody saying, oh, this particular bird has mutated and has a much larger breast? We're going to, use the genetic traits of this one for the rest of our family. We're going to breed that one, right?
Yeah. This is pure selective breeding. This is, I like the shape of that one. This one's bigger. Put that one in with the rooster. Let's see what we get.
Yeah.
Over and over and over again. Crossbreeding, inbreeding. Wow. And deliberately. And so we've made a Frankenbird. No two ways about it. And the other thing to think about is we don't actually grow. The modern fryer chicken is not alive for 60 days because you don't want an 8-pound bird. It's too big.
Yeah.
Right? You really want a 4-pound bird. So the reality is the average fryer chicken lives 6 weeks.
Wow.
And so really, if you think about it, what we've done now is turned that chicken into a meat growing machine.
Right.
We feed it grain and antibiotics because it's living so close to its neighbors that it gets sick a lot. And then after 6 weeks, we've even bred it so the feathers come out easily. So we can more mechanically remove the feathers from them efficiently, right?
Yeah.
This is all in the name of reducing food cost. A chicken in every pot, they said during the Depression. That was the goal, a chicken in every pot.
So there's some scary stuff that you just talked about right there that most people will say, oh my God, how can I eat, meat that doesn't have antibiotics in it? And I mean, forget about the GMO. We haven't even gotten to that point. But if you read, the Omnivore's Dilemma.
We don't need to get there.
But if you've read the Omnivore's Dilemma or any of those books or know about the industrial food complex, you know that they're squirting growth hormone into these cows and chickens and feeding them antibiotics like crazy, which is also causing antibiotic resistance, which is almost epidemic in terms of losing their effectiveness.
So I think that part of the outcry against GMO is really an outcry against industrial food production. For the most part, we don't need to know about our food production anymore. But if you understand that chicken manufacturing has gotten to a very horrific state without any direct genetic modification except for crossbreeding, you understand that you're conjoining two issues there.
Right.
Okay. And as people who are not happy with this, we have pressed against our lawmakers to make laws to make the chickens' lives a little less miserable.
And there are some chickens and some meat that you can buy that are supposedly free range and do not have antibiotics and all that stuff, but you pay at the grocery line.
Well, that's the trade, right?
Right.
Agriculturalists are not trying to torture animals. That's not the goal.
Right.
Right? The goal was to produce more food for less money.
Right.
And that has been accomplished. What's happened now is we have the luxury to look back and say, at what price?
Yeah.
And start discussing, am I prepared to pay more for my food to decrease the suffering of animals and to increase the quality of that food? Right? Not every chicken breast is the same. How it was raised matters. What it was fed matters. The outcry against the cholesterol in eggs had much to do with what was being fed to the chicken as it did with our understanding of cholesterol.
Right, which has changed a lot since then,
Absolutely.
Obviously.
The whole BSD event, right? This mad cow disease came from feeding cow to cow, which turns out to be a really bad idea.
Not a good idea.
Because we have an agricultural system that commoditizes the product, protein is protein, we don't make that distinction. And as we've learned more, again, don't think anybody set out to make mad cow disease.
Well, there's probably a good reason, evolutionarily speaking, why the idea of cannibalism is repugnant to us.
Yes.
And it may not have to do with our moral compass. It may have to lie. The answer may be more biological.
So once you get this idea that we have been manipulating the genetics of animals and plants for a long, long time, and that industrialization just got us better at it, going faster with it, then you can start looking at what are the new things we're doing, right? And again, I don't believe that we're striking out against GMO rationally. All too often, the conversation about antigenetic modification looks an awful lot like the conversation about anti-climate change and anti-vaxxing.
Or any of those.
They're all that same sort of alarmist mentality.
Right, because they do take some sophistication to understand in enough detail so that you can make a decision sanely.
Right. So being appalled with what we've done with agriculture so far is one thing. Let me tell you about some actual scary things. Were you ready for some scary things?
Well, I was not necessarily looking forward to it, but I was expecting some scary things in this show, Richard.
Right. So in the 1910s and 1920s, when we started understanding the structure of the atom and started understanding the electromagnetic spectrum in bigger detail, and nuclear options became a possibility, we started experimenting with using X-rays and gamma rays to create genetic diversity, right? Now, you think about one of the reasons we got so good at making chickens bigger and bigger is that their rate of growth is incredibly fast, right? When you can get a generation every 30 days, right? Maturity in 90 days, you can get a lot of generations going. So you can breed them quickly to get what you want quickly. But a plant often takes too long. It takes at least a year to see what the seed looks like or what the fruit looks like. So you can't breed them as quickly to be able to do experimentation. And if you're just counting on natural mutation, which in the end, quote, natural mutation means still hit with gamma rays, right? They're just coming from outside the planet.
Right.
So the fact that we use gamma rays to accelerate that process doesn't seem all that weird now, does it?
Right.
We've used chemicals and high energy particles to do damage to genetic material in plants and then grown the results to see what we got.
Yeah, right.
We have since the 1930s. It's a normal thing. In fact, I would argue that every single foodstuff made today out of plants probably has mutagenic genes from this process.
So do you call it irradiation? Is that what it is?
Now, typically irradiation is talked about as a different thing. That's for sterilizing food.
Okay.
But mutagenic organisms, and I've just sent you a link and I'll add this to the show notes as well.
Okay.
This is the IAEA, right? This is the main agricultural site for the world. This is their mutant variety database. So this is a collection of all the mutated food that is considered safe to eat. Would you like to know how many variations there are currently in the database?
Well, who considers them safe?
These are the experts that study them.
The experts.
Right? Yeah.
I want to know what their incentives are.
Well, now you understand this has been going on for 80 years, right? If it was going to kill us, it probably would have.
Maybe it is.
Right? But the reality is we count on mutation. We find feature. We look at the mutants and the new feature they've got. We like that. It's a benefit in some way, right? Yeah. Maybe it's more cold resistant, right? There's a more cold resistant version of rice in that database that was created by a gamma ray exposure. Right? Yeah. They even have the documentation about how they made that particular mutation. Now, once they've done that initial mutation, they will then breed it with other plants to get it to grow stably. Right? They're looking for a particular feature and they'll breed it over and over again with you. So you got a rice that you've successfully mutated with the gamma radiation. This is long before you could look at its genes in any way. But you can see its feature when it grows. And now you incorporate that into your breeding plan, breed the plant over and over again until you get a consistent growing version. So this is the World Food Organization responsible for cataloging all of this information. Almost everything we've eaten is mutated in some way.
Wow.
That is completely normal. This is considered organic. None of this falls into GMO in any way.
It's considered organic for other reasons, though, right? I guess the organic label doesn't include this mutation stuff.
Not at all. Organic is about your growing practices, not your breeding practices. Right? And that gets us to a very important point in this whole conversation, which is that genetically modified organism, GMO, is a legal term, not a food term. It's completely a legal concept. When we got concerned with what professional agriculturalists and the industrial agriculture machine was doing to food, we asked our lawmakers to put rules around it. And so they had to come up with legal terms for those things. That's how this works.
Wow. That is scary.
Be scared because in the end, everything I've talked about is pretty much natural. Gamma rays hit us every day.
Yep.
Gamma rays hit plants every day.
Yeah.
And sometimes the plants repair the genetic modification. Sometimes they mutate to it. For the most part, if the mutation is too severe, the plant dies off. Right? So when you realize that this is how plants have always evolved, all we've done is accelerated. It's not actually something to be afraid of.
Except for one little point.
What's that?
Do we trust the people with their fingers on the modification genes?
Now, we don't have fingers on the modification genes at this point. It's random, right? We're randomly damaging genes. We're looking at which ones grow, seeing if they have features that we like, and we're keeping them. Okay?
So we're in 3 levels of acceleration, right? First one is grafting and selective breeding. The second one is this sort of mutation observation, which speeds it up. And the third one is genetically modifying. And this is where, that ethical question comes in because, if companies are out there patenting genes and processes for making things differently and better and very quickly, obviously, and things like CRISPR make that ridiculously cheap and easy. Do we trust them? How can we keep our eyes on these people?
Well, let's talk about what we're really doing when we're genetically modifying food, because we're not doing it for fun, right? I mean, there's obvious things that we're trying to get to.
Sure.
We're trying to expand the range of the plant. Can it grow in colder environments? Can it grow in wetter environments, drier environments, hotter environments? I mean, that, and we need to do that because we're messing with the climate of the planet, and we're going to need to be able to move our food around.
And fundamentally, we want to increase our yield, right?
Right. We're trying to increase yield. We're trying to improve the nutritional value of the food.
Right.
We're trying to protect the food from being damaged by pests, whether that is insects or weeds or disease. They're all things we're trying to add in these capabilities one way or the other.
Okay.
And again, we've been doing this for a long time. We're just getting better at it. As we started to understand the way that plants actually function, and that's the thing that's interesting when you really study gene modifications. Gene modification came out of research into understanding how plants really worked, how they grew their fruit, how they made their seeds, what that all looked like. And once we understood it better, it's like, hey, that means if we made an alteration here, we could actually improve things. So the core mechanism up until very recently, actually still really the core mechanism. Every GMO plant you know of today, including all of the ones with the glyphosate and so forth, which we're clearly going to carry to another show, were modified more or less the same way with agrobacterium.
Okay.
So agrobacterium is a particular bacterium. This is the tumefaciens version of it.
Okay.
And it is absolutely a normal, natural process to do genetic modification. We call it a virus. Right?
Yeah.
That's why we started calling software viruses, because what viruses do is they inject new genetic material into cells.
Right.
And so what happened is, as we understood that process, we realized, hey, we can take this agrobacterium and we can breed it to get a particular type of plant cell into it that we want in our plant, whatever that may be.
Yeah.
Okay. So in the case of making Roundup ready crops, Monsanto figured out, they found a plasmid, basically a bit of genetic material in a plant that was resistant to glyphosate. And so they got that plasmid into agrobacterium, which they can insert into other plants.
Now, you just said a whole bunch of stuff that we probably need to unwrap. Right. One of them was Roundup Ready. Now, what is Roundup Ready? And for that matter, what is Roundup?
So Roundup is the herbicide that is most people are upset about these days when it comes to GMO. This is Monsanto's most profitable product. I will literally do a whole show on it next because it's such a huge topic. But
the basic idea is that Roundup is something that kills all the weeds and leaves the plants intact because they've been genetically modified to resist
be resistant to it.
Yeah.
That's right. And the mechanism they use, goes back into the 90s. So this has got nothing to do with really fancy genetic modification. This is a pretty basic one. This is taking a normal bacteriological process of virus insertion, only now they're picking the plasmid, the kind of virus they're inserting into the plant. And by the way, it's not particularly reliable. Typical time to make a genetic modification this way and create a reliable plant, 5 to 10 years. Probably $100 million. Wow. That's why only a handful of companies do it.
Wow.
It's really hard to do.
And could they use something new like CRISPR-Cas9 if that process got more standardized?
Absolutely. They're very interested in doing that. Now, I mean, that in terms of production crops, that approach, the agrobacterium approach, is the norm. There's a couple of new approaches. So one is called gene guns. So now we take the gene, the same way which we'd normally try and incorporate into a plasmid, and we actually put it in these tiny pellets coated in gold, and we insert them using a gene gun into a plant. The gold naturally dissolves, and sometimes the gene actually adheres to a plant DNA. Now, this is often where you could say we're going wrong, okay?
So it adheres to a plant DNA. Does that mean it spreads to all the cells?
Well, it'll spread to one cell, but if that cell is the one that reproduces, that's good enough. And you're not firing one pellet. Think more shotgun. You're firing many, many pellets.
I see.
Trying to get that particular plasmid to grow rapidly. Okay.
All right.
So again, it's only so precise. But the difference between agrobacterium and the gene gun approach is this. Agrobacterium can carry plant DNA fairly well, but it can't carry other DNA particularly well.
Okay.
So one of the science fiction kind of terrifying things you may have seen on the news is when they made like rabbits that glow in the dark. By taking jellyfish genes and putting them into rabbits.
The phosphorescent qualities of jellyfish. Yeah, that's a sort of a genetic parlor trick that's going around right now.
Yes. And believe me, none of this stuff is in production. You can't buy a glow in the dark rabbit, right? But it is experimentation is going on, and these new techniques allow us to take virtually any kind of genetic material that is interesting to us and insert it into other genetic material. All right. Agrobacterium sort of naturally confines itself. You can take plant genes and put them on other plant genes. So in a way, we're doing more precise crossbreeding. Right. And you remember going back to our original agricultural show, bred wheat came from, we've now seen from genetic analysis, einkorn wheat crossbreeding with goat grass. Because they were planted side by side, they naturally, eventually, somehow the genetics blended and you got this new kind of wheat. Right? Now we finally have tools, but, so agrobacterium, we could have done that faster. Because you say, hey, I like this nature of goat grass and its softness, and I'll take this plasmid, I'll put it into another plant. So agrobacterium tends to go plant to plant. That's fine. But the newer techniques now, we can pretty much take genes from anything. And that's where you get into sort of the ethical elements of this.
Yeah, okay.
Right? When you think about the radiation approaches, the mutagenic approaches, all you're doing is smashing existing genes. You're not adding new ones. You're just smashing them. And that sort of that's where the CRISPR conversation starts out.
Right.
Because CRISPR is a huge topic all by itself, and maybe we'll end up doing a show on it.
Well, we'll put a link to it anyway. It's a new process by which you can very cheaply and reliably edit genes and replace one set of gene sequence with another one, and it's having a huge impact across the spectrum.
And it's not necessarily that cheap or simple. It's just cheaper and simple than what they were doing. Every other method, including the gene gun method, has a randomness to it. You're just firing those genes at the cells and hoping for the best.
Yeah.
The original CRISPR mechanism was really about removing genes, right? It was developed to do research into understanding genetics better.
Yeah.
They were trying to identify which particular gene is responsible for which particular trait. And they discovered a totally natural process, and this is the Cas9 enzyme that allows you to that is actually built to fight viruses, so that when viruses modify genes, this is the mechanism that puts them back.
Yeah.
But it normally uses RNA that's already in the cell. So it already has another copy of pieces of its DNA so that it can repair itself. But CRISPR by itself will just do the cuts. It'll just take a little bit of genetic material out. And if you think about it, if you use CRISPR like that, it's not that different from the gamma ray approach. It's more precise. Right. But in the end, all you're doing is smashing an existing genetic material. But when you combine this entire mechanism, we can now take pretty much any gene we want and edit it in.
Right.
The one other big difference with the CRISPR approach is that I can do multiple gene edits simultaneously.
Interesting.
In every other mechanism. So you think about it. I'm trying to add to a plant resistance to a fungus. Okay. And this is actually a problem going on today with the banana, right?
Yep.
The Cavendish banana is dying out from the same blight that wiped out the Gros Michel banana in the 50s. The blight is evolved. It's now attacking the new banana that was the replacement. We've tried all kinds of things to try and fix it. And one of the things that genetic analysis has done to this fungus is realized it's hitting the plant in a lot of different ways. There's not one simple way to stop it. And with normal gene gun editing or even agrobacterium editing, you could introduce a trait to a plant, but it takes years to get it consistently breeding.
Yeah.
Right? Because of all these natural defenses that biological systems has for genetic modification. To get a modification to take, you have to try again and again and again until it's actually stable. In some ways, that's the validation mechanism that makes genetic modification safe. If it grows and reproduces reliably, in many ways, it's most likely safe.
So CRISPR/Cas9 has a chance here because they can put together a sort of cocktail of gene edits. Where all the places where they think it might need all at the same time. So boom, your genetics have changed.
It's a much more powerful tool.
Yeah.
Right? So if we could figure out the 4 genes involved in the penetration mechanism of the fungus into the plant, that we could make those 4 edits essentially in one pass and then breed that plant to stability, it would take less time to make those changes. You think it takes several years to make one change. So if I have to make 5 changes, now it's many, many years. But what if I could make all 5 changes and then it's one iteration of stability? That's what's interesting with that.
Do you think that we as a species have gotten sort of complacent with just the amount of good food and the avoidance of problems that have typically ravaged our food supply throughout the years?
I think that's part of it. And this is also where you get into the anti-vaxxer movement. We've had so little measles for so long, we're just not afraid of it anymore. Right? We've had this swing from everything needs to be clean, although we're still obsessed with cleanliness, to, white bread is bad. Right? White bread was originally the future bread. It was the better bread. Right? We've had these oscillations.
It was wonderful.
It was wonderful. Exactly. So let me talk you through, as we're running out of time here, a few GMO products. The original GMO product that falls into the legal classification of GMO using the agroplasmid approach was the Flavor Saver tomato. Did you ever hear that?
Well, I knew tomatoes were genetically modified, and they have been for a long time, but I didn't ever hear that term particularly.
Flavor Saver was a brand. It only was around for a few years, 3 or 4 years is all that the company lasted. The company was called Calgene, and they were a genetic company, right? Okay. They didn't know a lot about selling tomatoes, but they'd figured out they understood the tomato better than anybody else, and they understood that how tomatoes actually ripen. What they realized was the ripening for flavor and the ripening for color and the ripening for texture are all different.
Wow.
Now, the problem with tomatoes is if you actually take them fully ripe and try and ship them, you'll destroy them because they're soft.
That's why people grow them at home.
Because they're delicious that way. Yeah. So normal tomatoes, and we could do a whole show on the grocery store process. Oh, yeah. Maybe we should. Let us know.
Definitely.
They ship them green. They have to.
Yep.
And that's why they taste like crap.
Yep. That's why they're mealy.
But the Flavor Saver, they figured out how to separate ripening the fruit so that it tastes good from softening the fruit. 2 different things. right? You can ship a tomato green, but people won't buy it green. They want it red. So you treat the tomato with ethylene. Ethylene makes the tomato turn red, but it doesn't ripen the tomato for taste.
Huh.
What the Flavor Saver tomato did was that they had suppressed the chemical compound that causes the tomato to soften so that you could fully ripen the tomato on the vine. In fact, it would even turn red, but it was still hard. So because it was still hard, you could ship it.
And then did it complete its process en route to the grocery store and in the store itself?
Yes. You actually when you got to the grocery store, you gave it this special spray that activated that compound that would naturally soften the tomato, and it would be soft.
Interesting.
So it'd be a normal, ripe tomato.
Was it safe?
The FDA approved it.
Right? Was it safe?
Is driving safe?
Yeah.
Is flying safe? Is breathing safe? Right? What they saw was we have not significantly modified this plant. All we've done is suppressed the ability for it to soften, and then we enable it later.
Yeah, okay.
Okay? And so that's why it got approved. This whole attitude that government organizations are incompetent is a recent thing.
Yeah.
For a long time, we trusted that we paid the best professionals in the world to actually understand this stuff. And we could have a whole conversation, and maybe we will, about how that process may have been subverted as well as how the public perceives it.
May have been. The FDA is one of the most corrupt government agencies we know.
Yeah, most corrupt is a hard concept, but we'll talk about that.
Well, it certainly is corrupt.
Well, is it? That's an interesting question. The interesting thing, of course, is the Flavor Saver's gone. You never guess who bought the company when it went bankrupt.
Monsanto.
Why, you'd be correct, sir.
Yes, I knew.
Yeah. Now, let me say something very nice about Monsanto, and I'll tell you another story. And this is the Hawaiian papaya.
Okay.
So in 1995, there were almost no papayas being grown in Hawaii anymore because of the ring spot virus.
Yep.
The ring spot virus was transmitted by insects, typically aphids, which are almost impossible to stop. But it was actually a virus that went into the plant, and it ruined it, it made it spotty and soft, and it would rot very quickly.
Uh-oh.
Okay. And so they tried selectively breeding that problem out. They tried crop rotation and discipline to get rid of the virus and so forth. They tried quarantining different plants. They did all of the least expensive solutions to try and solve it because genetic modification is extremely expensive. So it wasn't until all other options had been sort of buried that they used genetic analysis to understand what this illness was, right? And realized that in the papaya DNA was susceptible to this particular virus modification that would ruin it. But there were other plants that were not susceptible to ring spot virus. And so government researchers actually said, if we could take a gene from the virus itself, known as the coat protein, and embed it into the skin of the papaya would be resistant to it. And it was actually Monsanto who did the work. They actually did the agrobacterium plasmid to actually figure that out, and then they gave it to the Hawaiian Farmers Association. Now, they said point blank that it's like, look, there's no money in it. There's not enough papaya for this to be a business we'd be interested in anyway. We know how to do this, so we'll do it. It got approved and so forth. We'll provide these modified seeds at cost, right? Knowing it would reproduce on its own anyway. And the reality of this is that this virus, which had been transmitted from insects for a long time, this is something that's been in food for a long time. It's not dangerous in any way. It doesn't affect people. It only makes the papaya go soft and rot, right? That's all ring spot virus does. But now it was a genetically modified food. And so it actually got attacked, right? When they started growing it in 1999, people started freaking out about it. Actually, there were vandals that destroyed cheese, calling it genetic pollution, recognizing if they didn't do this, there would be no papaya at all, right? And there was a paper produced in 2002 that said there are parts of the gene in the ring spot virus that look like an allergen, right? But there was no there was no complete study that actually said, here's how you trigger the allergy and so forth. But they used that study to attack it for further on. There were 6 modified amino acids of the 280 in the papaya gene. That was the modification that they made, right? And eventually people lost interest in this. It was only the papaya. People stopped caring. Japan approved it in 2011, started growing it there as well because the ring spot virus has been spreading around the world. So this is now like the alternative, the only reason papaya still exists. is this now modified version.
Wow.
Right? So there's no money in it from Monsanto, even though they did the work. The research was originally done by public sector scientists anyway. It doesn't cross-pollinate to other plants, so it's not really affecting anything. And more importantly, because the plant's now immune to ring spot, you don't spray pesticides on it anymore to try and stop it. Right? They used to try and kill all the aphids to stop ring spot, but aphids are so plentiful, you couldn't use enough pesticide.
Wow.
So it's one of these examples of genetic modification that benefited everybody in the end.
Yeah.
Right? And it looks like it's the only chance for us to say the Cavendish banana as well. We really don't have any other option.
Right. Well, it's be interesting.
Two more.
It'll be really interesting to follow that story. I learned about CRISPR-Cas9 from Radiolab, and we've talked about it before. And I'm always watching the news to see how that plays out.
Well, and at this point, there have been no products made using CRISPR.
Right.
So it's just a research piece, and this is part of the challenge I think most people have. I think the scariest time for mutating crops and so forth, genetic modification, was probably in the 50s and 60s with the sheer amount of mutagenic behavior that was going on and only a marginal understanding of what was going on. Now that we know so much more and we're much more precise in what we're doing, it's far less threatening.
Yeah.
Have you heard of golden rice?
Tell me.
So golden rice is genetically modified rice. Now, why are they modifying the genetics of rice? Vitamin A deficiency. So in Southeast Asia, it is hard to get enough vitamin A to keep yourself healthy, actually. It's a real battle. In fact, hundreds of thousands of children go blind because of vitamin A deficiency. It's considered a major health crisis, and the Office of Humanitarian Services in the UN is spending a lot of money to give kids vitamin A in the form of liquid a couple of times a year so that they get enough vitamin A to maintain their sight. Because they just don't have access to beta carotene the way we do. These are poor people, and their primary staple crop is rice. And so there was this pretty good idea back in 1999 to start incorporating beta carotene into rice. Now, again, they were using that agrobacterium, so they were only using genes from other plants. Daffodils, typically, was the initial thing. And it didn't put enough beta carotene in the plant to actually make a difference. There was, again, no money involved. This was pure government science trying to save lives. It got attacked because it was genetically modified. Of course it did. And it was a double whammy that it wasn't enough beta carotene to make a difference. But by 2005, they'd actually raised, by doing additional crossbreeding and additional genetic modifications, they'd raised the beta carotene by 20 times.
Wow.
So that now, there was a Chinese study done in 2008 that said a 50-gram serving of the golden rice would give a child 60% of their daily intake of vitamin A.
Wow, that's amazing.
It's huge. And yet, there are organizations that continuously attack this product so that it's still not available to most people. The UN can only afford to go in and give children these doses of vitamin A twice a year, which is not good for them. They still have sight problems.
Right.
But they really should be getting vitamin A every day. This is a good solution to the problem, but because it's called GMO, it's just attacked.
Well, this gets me back to the thing I said earlier, which is, and I think people's fear of GMO is that it seems like a binary decision, either yes or no. You either allow all GMO food in or you don't. And while we don't have a fear of things that take more time, thought, money, and resources to modify, right? Like is the way we've been doing it so far. The fact that a company like Monsanto with their finger on the CRISPR gun could just use this to boost their own sales or push food on people or bully farmers into growing food that is beneficial to them, not necessarily beneficial to us. I think that's what worries people about GMO. Am I off base there?
No, I think that a lot of people are worried about that. There's just no actual evidence.
There's no actual evidence of what?
Of any of these things actually happening. Genetically modified food take a long time to make.
But are you saying there's no evidence that companies like Monsanto will modify the genes of their seeds for their own benefit?
Well, they absolutely will. And I'll do a whole show on that when we get there. But you always do stuff for your own benefit. The question is, does it benefit other people, right? Is it a win-win?
Right.
In the end, the primary benefit to the farmer is they grow more food for less money, right? And does Monsanto deserve to make a profit from that?
Well, sure it does. But one of the things they do is they make seeds that you can't reharvest. In other words, you have to buy seed from Monsanto every year.
So that turns out to not be true.
Really?
So the Terminator gene concept, which is the idea that the seeds will be sterile, never made it out of the patent office.
No kidding.
Okay. That being said, virtually every farmer buys seed.
Every year?
Every year.
Okay.
Because it grows more food. The process of growing seed is different from growing food.
All right, I got you.
Right? So you can plant the seeds from your previous crop. You'll just get less yield from them.
Well, that's great to know, Richard. And this is why we have these conversations.
Right. It's a totally normal process in modern agriculture to grow seed separately from food.
Okay.
Because the process of growing seed to get that high yield is different from growing it for food.
All right. So in all of your research, you're saying you haven't found any evidence that Monsanto has done anything that isn't also good for its customers.
Not true. I absolutely have found issues with all of that.
Oh.
They have totally run afoul of the patent process. But understand, that's not them trying to kill farmers. That's not the business they're actually in, right? That's them. The patent law is clear. If you don't protect your patent, you don't have your patent.
Yeah, right.
Right? So and you combine that with a legal system in the United States specifically that is largely built around bullying.
Yep.
Right? Forcing settlement rather than actual negotiation.
Yep.
And you get the situations that you see. And I'm not saying Monsanto is blameless in any stretch of the imagination.
But in the environment that they're in legally, which I agree is ridiculous, that happens.
Right. And on top of that is this issue of patents do expire. So the patent on glyphosate has expired. Anybody can make glyphosate now. Right? So how does Monsanto continue to make money from that product? With their genetically modified seeds.
I see.
Right? That's the product they can still protect. That's the product they can continue to sell.
Okay.
They can't protect the herbicide anymore, so they're trying to protect the plant. And that gets us into a whole conversation, which again, might be its own show. People have to tell us if they want to do this, about the problems we have on patenting genetics. Right? Really saying, if you learn something about a natural process on how a plant grows, do you deserve a patent on that? And if I don't think we have a whole lot of problem with if you modify a plant a particular way, you're allowed to sell that plant. The question is, if somebody else makes the same modification and came to it their own way, their own route, is that actually distinct stuff? Can you stop someone because they've made the same modification by discovering the same things? Right? If you learn that this modification in a gene will cause this plant to grow twice as much, that's actually a natural process. It's that modification. It's the only one. It's unique. You didn't invent it. You found it.
Yeah.
Do you deserve to keep it to yourself?
Okay.
So what I have planned next is a show specifically. So if you think about all the stories I've told here about genetic modification, they're mostly about increasing the yield on food, protecting food from disease, extending the nutritional value of food. All fairly positive things. Yeah. But it's not the only thing that we modify for. The pesticide resistant side and so forth, and this is the thing people are most sensitive about, is a huge topic. And it's why I've spun it off as its own show. We're going to talk about pesticide resistant crops in the next show. But I can think of several others we could go into here. So I'm happy to have folks send us emails or write to us on the show notes itself about what they'd like to hear next. I'm also thinking, how long do we want to talk about agriculture? Because there's some cool stuff going on in space these days,
there is,
too,
isn't there?
that I wouldn't mind talking about as well.
Yeah, awesome, Richard. Well, this just whets my appetite for the next one, man.
I love it.
Yeah. Thanks for listening.